question. So let me show you exactly as we are comparing the question versus where do I find the answer in the booklet. For this case, I will only focus on questions not related to objective. So on the side of the objective, I'm going to be a little bit faster. Let's start with the section A. The component of DNA molecule that provide the code for protein synthesis. Which component of DNA molecule that provide the code for protein synthesis? So what causes this are the nitrogenous bases. What are the nitrogenous bases? We are talking about the thymine, the guanine, the cytosine, and the adenine. These are the nitrogenous bases which are supposed to be organized together to form a gene. So these are the ones which bring about a protein. So the answer is D. Then 1.1.2, during which stage of meiosis do spindle fiber begin to form? So the spindle fiber always begin to form under the preparation phase, which we call the prophase. So it means that the answer is going to be A. I'll explain more when we go to other sections. Then they're saying that an individual that has Down syndrome in the karyotype, there is an abnormal number of chromosome at chromosome pair. So Down syndrome, whenever you talk about Down syndrome, yes, so your mind must go to chromosome number 21. Whenever you talk about Down syndrome, so you must think about an extra chromosome on chromosome number or chromosome pair number 21. So the answer is going to be C. So don't forget that these objectives, each objective is 2 marks. Then they're saying that an individual that receives an identical allele from each parent is described as, how do you describe an individual that receives an allele which is identical from both parents? For example, you have the parent is capital T, small t, and then another parent is capital T, small t. So now, if I receive this and this, how do I call this? So now, if the baby gets small t, or this and this, and the kid is capital T, for example, yes, it means that these 2 alleles are the same. So how do we call that individual which receives those 2 alleles? So because they are the same is homo. So it is going to become homozygous, because we are describing a zygote, yes. So that zygote has the same kind of allele. So the answer is going to be C, meaning that homozygous. Then the next question, they are saying that the plant species that has a diploid chromosome number of 12. So if a plant species has a diploid chromosome number of 12, yes, which of the following is a haploid chromosome number for this species? It means that if it diploid, di means 2. Di means if it is diploid, it means 2. That's why it's called 2 sets of chromosome, 2n. So haploid, diploid means 2, and then haploid means 1. If the diploid is 12, and this means 2 sets of chromosome, then haploid means half. Yes, so half of the 12 is 6. So 1n is going to be what? Is going to be 6. So when we come back here, we shall see that the number of chromosome from 12 is going to be, half of 12 is going to be 6. Yes. So it's going to be 6. So the answer is going to be C. Then they are saying that inheritance by multiple alleles. Inheritance by multiple alleles in genetics is referred to as 2 alleles that influence the 2 characteristics. 2 alleles, no. It's more than 2. More than 2 alleles, this is correct, that influence 1 character. Yes, it is more than 2 alleles. Then those 2 alleles must influence those 2 alleles must influence 1 characteristic. A good example for this is blood group. Yeah, it is 1 allele, it is 1 gene. Which gene is blood group? That's 1 character. And then, how many alleles are there? We have allele for A, dominant allele for B, dominant allele, and then allele for O, recessive allele. So we have how many alleles? There are 3. So because there are 3, they are multiple. These multiple alleles will influence 1 gene or 1 characteristic, which is blood group. So that's why you're saying that B is the most correct answer. More than 2 alleles that influence 1 characteristic. Which one of the following is a reproductive isolation? When you talk about reproductive isolation, it means that the organisms are being isolated as a result of the reproductive strategy or the reproductive mechanism. So they're saying that breeding at the same time of the year, they can't be isolated if they are the same time of the year. Infertile offspring, this one is correct. Plant adaptation to the same. If it is the same pollinating agent, then they can't be isolated. Improved fertilization, this just increase the ability to bring about fertilization. So the answer is going to be B. Why? An example is, for example, a horse. You have a horse and then a donkey. When you cross the 2, you've produced a male which cannot reproduce again. It is infertile. So it means that they are being separated. So the answer is B. Then you're saying that normal human ova have normal human. Remember that when you talk about humans, they have what you call 46 chromosomes. Yes, but each ova or sperm, it has half of the chromosome number. Yes, so it means that it has 23 here, 23. But among these 23, if it is a male, it's gonna have Y, and if it is a female, it's gonna have X. But which chromosome number will have the Y and which chromosome number is going to have the X? The chromosome number 20, the 23rd chromosome is going to be X on the female, and the 23rd chromosome is going to be Y. 23rd is going to be Y on the male. So that if now the Y of the male fuses with the ovum of the female, which is X, is going to be XY, and then we produce a male. And then the female is always X, so that's why the X of the male fuses the X with the X of the female, then it's going to become XX, and then it's going to become a female. So now they're saying that normal human ova have 22 autosomes. From chromosome number 1 until chromosome number 22, we call them autosomes in both male and female. So 22 autosomes. So whenever I have 22, that answer could be correct. I have a 22 here, I have a 22 here. So it means that now these 2 answers are wrong. So we have wiped them out. So it means that I have this and this. But they're saying a normal human ova. When they talk about ova, they are talking about a female. So this must go to female. Yes. If it is a female, female can't have Y. They're always having X. So it means that the answer is going to be X. And 22 autosomes and an X. It can't be 22 autosomes. Yes, it's 22 autosomes and a Y. Then it is going to become a male. It's going to become a sperm. So it is 22 autosomes. So basically it is an ova. It is not a sperm. Then the next question is saying that which one of the following occurs in mitosis, but not in which one of the following occurs in mitosis, but not in meiosis? When you talk about mitosis, you have one cell. You have one cell. One cell will divide to form two cells, but these two cells are identical. But if you look at meiosis, you have one cell, you form two cells in the first meiotic division, you divide again, you form four cells. These are the one which forms the sperms and the ova. And then these have the chromosome number. They have half the chromosome number to the parent, which is, for example, if it is a human, is 46, and then here will experience 23 chromosomes. So now what happens here in the question? They're saying that two cells are formed at the end of the division. They're saying which one of the following occurs in mitosis. So it means that it occurs, we are comparing mitosis over meiosis. It means that the first statement must be belonging to mitosis. The second statement must be belonging to meiosis. So the first statement, they are saying that mitosis, they're saying two cells are formed under mitosis at the end of the division. Yes, we see here two cells are forming at the end of the division. They're saying crossing over takes place. We don't have crossing over in mitosis. We don't have crossing over. So this one is ruled out. The most correct answer is A. So the answer is going to be A. And then the last objective is about the graph. Is about the phylogenetic tree. They're saying that the graph below shows the percentage similarities between humans. So we are not talking about differences. We are talking about similarities. Percentage similarities between humans, and then DNA, and then DNA of some species of African apes. So it means that if you look at this, if it is similarities, it means that humans and the chimpanzee will share 98% of DNA. And then gibbon and the humans, they share 96% DNA. And then the rhesus monkey, we share 94% DNA. And then the galago, we share 72% DNA. So now a question is asking, which one of the following phylogenetic tree best represents information in the graph? They are saying that basically if you look at these percentages, the more is shooting towards 100, the more is like we are related. It means that humans and the chimpanzee, we are more closely related than humans and the gibbon. Why? Because the difference here is 2, while the difference here is 4. And then the difference here is 6. It means that we are more closely related. If we look at diversion, we diverted recently compared to the gibbon. Now let's go to, we are seeing here human and chimpanzee. Yes, they are a little bit closely related and they are recent. If you look at them here, 2% difference and 4% difference. And then it means that we are recently, but we are next. The gibbon, chimpanzee, yes. The gibbon diverted after. No. Before the chimpanzee diverted from human. This is what it means, that the gibbon the gibbon diverted before the chimpanzee did what? Did separated, and then the rhesus monkey, because it is 6%, was the first before the gibbon separated, and then if you look at the galago, separated a long time ago. That's why you see that the difference between this and the human is too big. Why? Because they diverted long time ago, so the nature modified them differently. So if look at the most correct answer will be C. You'll find out that the most correct answer is C. This is a phylogenetic tree which we are requires to be studied a little bit more. And if you look at it, you don't need to cram, you just have to know the principles of understanding of the principles of the phylogenetic tree. How do you apply this phylogenetic tree? Basically, just have to look at the similarities and differences. Then these are 10 questions. In most cases, they come between 8 to 10, and each question comes 2 marks. So now we're going to go to question 1.2, and this is related to the terminologies. 1.2, which are biological terms. Here is saying that the division of the cytoplasm. Now here we are going to see the answer corresponding where we are supposed to get these answers from our booklet. As we said that it is a distinction material, so we need to find out each and every question which is in this paper, where are we going to find the answer. This division of the cytoplasm, this is called cytokinesis. So where do we find this? How do we know this? So we're gonna go to our book, and then you go to meiosis. Yeah, it's here, telophase. We are saying that the daughter cell chromosomes reach the pole, a new nucleus form, the cell membrane of each cell constrict. The cytoplasm divide into cells, which we call cytokinesis. So meaning that division of the cytoplasm is called cytokinesis. Here, that's how it is being answered. And then we go to the next question, the sugar molecule present in the nucleotide of RNA. It means that the kind of sugar which is found there is ribose. So where do we find this? We go back to our book on DNA, we're gonna find it here. You're saying that single-stranded molecule consists of nucleotides. Each nucleotide is made up of a sugar called ribose. So you see the answer is there. And this is the structure of RNA. So ribose is the sugar which is located in RNA. The position of a chromosome, the position of the gene on a chromosome, this one is called locus. So if you go to our book, where do we find the position of a chromosome? We'll find it at the beginning of genetics, position of the gene on the chromosome. So basically, these are what we call the locus. And also here, you see that these are positions of genes. So each individual position is called a locus. And if there are many, we call them loci. Another question, they're saying that the process by which DNA makes exact copy of itself, this is DNA. This is what? DNA. We'll find out that it's called DNA replication. I think this one is easy. You can find it in DNA replication. What is DNA replication? Is the process by which DNA makes as an exact copy of itself. And if you look at this question, they are saying the process by which DNA makes exact copy of itself. So exactly what is there in that book is just what they brought here, and then they're asking you the question. So it is DNA replication. Undifferentiated cell that may be stimulated to develop into any body cell. So we call these ones stem cells. So where do we find this? We'll find this in our book in genetic engineering. Yeah, there we go. You're saying that stem cells, these are cells with unique ability to develop into specialized cell type in the body. So if you look at the question here, it's saying that undifferentiated cells that may be stimulated to develop into body, into type of body cell. So here you're saying that these cells, what happens? They can develop into specialized cell types in the body. So it's just a reverse, but that's what they are looking for, which you call the stem cells. So there is no question which you're going to find in the booklet, you're going to find in the exam, and then it's only being answered by this booklet, which we call a distinction material. Excel in life science. So now there is another question. They're saying that Mendel's principle, which states that the organisms possesses two factors which separate that each gamete contain only one of these factors. Why do we call them factors? Before we started to call them genes, we are calling them factors. The answer here is Mendel's law of segregation. So basically, that is the answer. It's a segregation. So now, where do we find this answer? We will find this answer from this booklet. Yeah, we'll find this answer on this book. Mendel's law of segregation states that, they're saying that Mendel's law of segregation says that the diploid organisms passes randomly selected alleles for a trait to its offspring, such that offspring receives one allele from each parent. So the question here is asking, the principle which states that organism possesses two factors which separate. Yes, they separate so that each gamete, that is the offspring, each gamete will contain only one of these factors. So here they are saying that the diploid, it means that the organism passes randomly. Yes, we don't know which one is it, but randomly, it happens randomly. The selected allele for a trait into offspring. How do you pass this? We pass them through gametes, such that the offspring receives one allele from each parent. So this is Mendel's law or principle of segregation. So our answer here comes back to Mendel's principle of segregation. Then another question, they're saying that evolutionary theory that propose long periods where species do not change, alternating with the short periods where rapid change occurs. This is punctuated equilibrium. Where do we find this? Yeah, the answer is here. It's saying that punctuated equilibrium, what happens to it? This evolution involves long periods of time where the species do not change. So it alternates with, it has short periods of time where there is a rapid change. There is evolution, there is long period of time where the species don't change, and it alternates with the short periods of time where the species is changing. So basically, long period of no change, and then short periods of species changing. If we come back to our question, we shall see that they're saying that theory that proposes that long periods of species not changing and alternating with the periods where the species is changing and it is short period. So basically, that is the answer. So this is also being answered by the book. Tangled network of DNA protein located in the nucleus. This is the chromatin network. Let's see where we find it. Yeah, we can find it here. Chromatin or chromatin network is being indicated here. Yeah, here is showing you that it's a substance within chromosome consists of DNA protein. So if you look at it, you see it that it is a network. It's a network. And also, if you go back to DNA replication, you'll find it again. It's chromatin network, how it is consists of. You see, these are what you call chromatin network. These are the chromatin network. So basically, still it can be answered by that. So you're saying that entangled network of DNA. So it means that it consists of DNA and protein located in the. So if you look at it, it is DNA, yes, and its network and is located in the nucleus. Then the natural shape of DNA is double helix. How does our book answer this? If you go to the structure of DNA, yeah, you have to go to the structure of DNA. Yes, describe the structure of DNA. You will find out that DNA strands twist to form a double helix. Forms a double helix. So basically, the book answers that question also. Then another one is the phase in cell cycle during which cell growth occurs. During which cell growth occurs. When does cell growth occurs? We shall see that cell growth occurs in interphase. So where do we find this in our book? So during this is what called the growth phase. And then we have the mitotic phase. So this is being answered by during interphase or before the cell division. So basically, if you look at DNA replication, that's when the cell is growing. It's supposed to grow. And then the growth occurs during interphase or before the cell starts to divide. So this can answer our question. A group of similar organisms that in a particular place at a particular time with ability to interbreed. What is it? A group of organisms that occur in a particular place at a particular time with ability to interbreed. Since it's a group of organisms, then it's gonna be and they're able to interbreed. Since they can interbreed, then automatically it is a species, but it's a group of these organisms. Then because it's a group, then it's going to become a population. So the word group cancels out for the species and then forms population. So the answer becomes only. And they're saying that manipulation of biological process to satisfy human needs. The answer is because it is manipulation, it is technology, but because it is biological process, then it's bio. So it becomes B only. Who discovered the structure of DNA molecule? James Watson and Francis Crick. Francis Crick, then it means that both A and B. Please always follow instructions because the instructions saying A only, B only, both A and B or none. Some people, they only write A, they write B, they write both. What is both? Both mangoes and oranges, both males and females. What is both? You have to say both A and B. Don't say A, because if you don't just say A only, it means that maybe even B is correct. What you do is you have to put only, both A and B or none. Try to follow the instructions. In tomato plant, tall stems are dominant over short stems, and the red fruits are dominant over yellow fruits. A farmer crosses a homozygous tall, then it means that now it is homozygous tall, so it means that it's going to be capital T with the yellow. Yellow is, we say that yellow is recessive, which is the moment they say yellow, I don't need to say homozygous or heterozygous. It's always recessive because it is always heterozygous. Sorry, it's always homozygous because it's recessive. Plant that is heterozygous for both characteristics. A farmer crosses homozygous tall, yellow, tomato plant with a plant that is heterozygous. It means that heterozygous for both, then it means that it's going to be capital T, small t, and then capital R, small r. Then name the type of genetic cross. Because you're involving two characteristics. Which characteristic? The height of this means that we are involving the height, and then crossing with the another characteristic is the tomato color. That is the color of tomato. So two characteristics, that is height and the color. So the answer is dihybrid cross. Where do we get it in our booklet? Yes, we get it from here. Dihybrid cross involve inheritance of two characteristics. So here we see that you have height and color, two characteristics. So we're saying that in the dihybrid cross, a cross which involve two character, and then they're saying give the genotype of homozygous tall and yellow plant. We have seen homozygous tall, we have seen it, which is capital T, and then yellow, which is recessive. We said it here is recessive, so it's going to be small. List the genotype of all of possible gametes for a plant with heterozygous both alleles. So if it means that if it is heterozygous, it's going to be capital T, small t, capital R, small r. So that because it is dihybrid, it means that I'm going to have this with this. It's going to be capital T, capital R, this with this. It's going to be capital T, small t. Then this with this. It's going to be small t, capital R. This with this. It's going to be small t, small t. That's why there are 4 marks, giving you this, giving you this, giving you that, and that. Yes. So we have this must go with these 2, and this must go with this 2. So this T with capital R, then capital T with small r, then small t with capital R, then small t, small r, small t, small r. Yes. So basically that is it. Then they're saying that in hemophilia, hemophilia is a sex-linked recessive, is a sex-linked trait. The pedigree diagram below shows the inheritance of the hemophilia in the family. Now, you always need to know that if it is sex-linked, even if they say male and female, but they have not mentioned that it's a sex-linked, please don't use X and Y. But if they say sex-linked, always use for a male, always put XY, while the female use XX. And you also need to know, before you go for this question, you need to know that always, because a circle, a box means a male, while a circle means a female. Sometimes we don't give you a key. Sometimes we can even ask you a box means what? Yes. So let's continue to our pedigree. Can this question be answered by our booklets? Yes, because we have many questions concerning about pedigree diagram. Let's see. Yeah, this is a sex-linked disorder. So it means that whenever you see a sex-linked, a box means a male, while a circle means a female. All right, let's go back to the question. State what is represented by a square or a box on the pedigree diagram. We have seen it that a square is male, while a circle is a female. State the number of generations represented in this diagram. You have to count them. 1, 2, 3. So those are 3. Those are 3 generations. 3 generations. Just count 1, 2, 3. Those lines. Then you're saying that offsprings of individual P and Q. P and Q. So these are offsprings. However, not whatever on this line means offspring. This gave rise to this, and this. What about this and this? This one is an intruder. who married this guy, married this girl from these people. And also this girl married this guy from these people. So these are just people who came into the family, but these are the offsprings of P and Q. So there are three of them. They are saying that give the letter only of female who have hemophilia. Letters of female who have hemophilia. So the shaded part is because it's recessive, they are affected with hemophilia. So now they are saying that give letters only of female who have hemophilia. So female is 1, is 2. So there are 2 people who have hemophilia. Then give the genotype of R. R. Where is R? R is a female, but a female came from a guy who had hemophilia. So it means that it is heterozygous and is a female. This one and this one, they gave rise to a baby who is sick with hemophilia. Then it means that the lady is heterozygous. So it means that R, since it is sex-linked, because it's sex-linked, they mentioned it. So I'm going to start with X and X because it is a female, and then hemophilia is caused by a recessive allele which is put on the X chromosome. So for normal, it's gonna be capital H because, and then small h. Why? Because this person is heterozygous, is a carrier who carries this recessive. Because we have to see how did they get this. If a male, if this guy is capital X, small capital X, capital H, because it is normal. Y at the moment, Y doesn't carry anything. And then this one X, capital small, then it's capital H, small h, so that now this combine with this, this combine with this, and then I form capital is X, small h, and then Y, which gives rise to this boy. Therefore, the female, if it was capital H, I wouldn't have produced the baby who is sick. So the genotype of R is capital H, small h. Then another question, this marks the end of section A. What about section B? What about section B? You see that the questions in section A, you can score all the marks. You can score all the marks. Question 2, they are saying that the diagram below shows or represents one cell in a phase of meiosis. This, they are not saying that this is a human cell. They have not talked about the word human. So you must not think about 23 pairs of chromosome, please. Don't talk about 46 chromosome, please, because they have not talked about human. So now they are saying identify the phase of meiosis. So they are in, they are along the equator, So because they're along the equator, so the phase is gonna be metaphase. But which metaphase? They are single, they are not double. If they had another one here, then they would have been double. So it would have been metaphase 1. But because they are single, therefore it is metaphase 2. So it means that the phase which is indicated there is metaphase 2. Give an observable reason for your answer because chromosomes have been aligned along the equator singly. So that it must show. Where do we find this in our book? We go to meiosis. Yeah, you see that the chromosomes are aligned along the equator singly. But if it was supposed to be meiosis 1, it would have been like this. They are 2, they're in pairs, but they're not in pairs, therefore it is meiosis 2. Then they're saying identify A. What is A? A is a cell membrane. Don't confuse the cell membrane with the nuclear membrane. What is outside is cell membrane. And then B, these are fibers which hold the chromosomes in place, which are spindle fibers or spindle threads. Then describe the role of B in the movement of chromosomes during meiosis. B, These ones, they are very important as they try to contract, they shorten, and then they pull the chromosomes or the chromatids to opposite poles. And also, they are very important in holding the chromosome in place, maybe during metaphase. So where do we get this? We get this during anaphase. Spindle fiber shorten and pull the homologous chromosome to opposite poles. That's where we get that answer from. What? Spindle fibers or threads are the one which hold these chromosomes. So the book is answering that question very well. Then another question that's saying that draw a diagram of draw a diagram of the draw a label diagram of the structure C that would appear in final phase of this mitotic division. Show the correct shading of C. So where is C? So remember, this is gonna split. It is a chromosome. It is a chromosome. So if it splits, let me draw it here. It is a chromosome, yes, with a centromere there, but half of it is shaded here. We see it. So when it splits, one is gonna go this side, one is gonna go that side. So it means that this chromosome is gonna look like that, and then the other one is gonna look like that with a shaded part. So what is this? When it reaches the pole, we call it chromosome. Chromosome. But which chromosome is this? Is unreplicated chromosome or single-stranded chromosome. Or we can call it daughter chromosomes. Yes. Whenever I ask you to draw, never ever draw anything without writing a title. You must always write a title in there or caption. The next question is about DNA. This DNA is basically under protein synthesis. So, can our book answer this question? Let's go there and see. Yeah, the question is there. That's where the question is. The question is here with us. It's there. And then here, there we go. They're saying that. Name the process that occur at W. This question. This is what they're looking for. So if you come here, they're looking for this. And then we also have the equation. We have this. This is the question. They're looking for what is happening inside this process. This process, if you look at this, what is happening inside the nucleus. That's what they are looking for, because this is the nucleus, which is transcription. Then they're saying, name the process. Why? Obviously, if you have transcription, this one is going to be translation. So they are looking for this, translation. Then they are saying that identify organelle X. Organelle X is this. They are looking for this organelle X, this wall. This wall. If you come here, they are looking for this wall, which is the nuclear membrane. Remember, remember, this is the nuclear membrane, the membrane of the nucleus, while this one is cell membrane. Yes. And then they are saying that molecule Z. Where is molecule Z? Molecule Z. They are looking for this. Because it is on the ribosome and it has moved out of the nucleus, is this. Then it is messenger RNA. Yes. And then they are saying that state 2 locations of DNA in a cell other than in the nucleus. They're saying 2. We find it in the mitochondria and also in the chloroplast. So let's see it in our book. If we come here on the types of DNA, we shall see that if you have the nuclear DNA found in the nucleus, they say other than this. So we have the mitochondrial DNA found in the mitochondria, and then the chloroplastic DNA found in the chloroplast, meaning that that is the answer. Then another question they're asking, describe the process W. What is process W? They're looking for transcription. How do I explain, or how do we explain transcription from our book? Transcription is being explained nicely. DNA double helix unwind. So basically, we give you a tick here. DNA double helix. Double helix, in most cases, we give you a tick. Yeah, DNA double helix unwind. So when you say unwind, we give you a tick here. The weaker region bond break. Weaker region bond breaking and unzipping is the same thing. One strand. This is very important. Sometimes they say two strand. No. One strand will act as a template mention of messenger RNA using the free messenger RNA nucleotide. Then they're saying that the messenger RNA is complementary. We like this, complementary to DNA, whereby A goes with U and then C goes with G. Remember, this is transcription. You are forming messenger RNA. So we don't have messenger RNA now has the coded message for protein synthesis, and then it moves out of the nucleus via the nuclear pore, and then it goes to the ribosome where it attaches itself. So you get the marks, get all the marks. The reason why this book is changed in different color, so that you know exactly where the marking point is. Yes. Then you're saying that another question is DNA-based triplet code for serine. Serine is here, serine, but they are saying this, they are looking for DNA. So now we have to convert this into two times, because we have tRNA, we have to change it to messenger RNA, and then we go to DNA. So if it is tRNA, it is saying serine is UCG, so it's gonna be U goes with A, C goes with the G, and then G goes with the C. So it's gonna be TCG. So that is the code for DNA. So it means that you have to convert it into two times. They're saying that the first two amino acids coded by the molecule Z in the diagram from left to right. The first two amino acids coded by the Z and the diagram from left to right. So left to right. So I have this, and I also have this. But I need to first read, this is messenger. I need to go to the table and check. So since it is transfer RNA, so I have to convert this into U goes with A goes with U, and then G. So and then another one is CAU. So AUG, I come here, I look for AUG, which is tyrosine, and then CAU, which is valine. What is the chance? What is the chance in the sequence of the nitrogenous base in DNA molecule called? What is the change in the sequence of nitrogenous bases in the DNA molecule called? This is gene mutation. So we call it gene mutation. Why? Because the bases have been changed. Then they're saying the code for this SCU on molecule Z changed to this. Explain the effect of this. This can also be answered by the book. If you go to the end of DNA, it is here. What will happen if this mutation changes the triplet 1 to this, or this to this? So basically, if you go here, then automatically it's being answered there. And then this is the permanent, is the mutation which is permanent alternation of DNA sequence that make up a gene. Question they have asked here, change in the sequence of nitrogenous base. How is it called? It is the gene mutation. So now, if this, we are saying that CUA, we are saying codon, so we have to change it into anticodons. So we are saying that CU is going to be GAA, and then this one's going to be GGA. If this codes for the same amino acid as this, there will be no change in the protein because it's just coded. It's like having two names, and I call you with the first name, you say yes, sir. I call you with the second name, still you say yes, sir. But if now I call you with a different name, you won't say yes, sir, because we're a different person. So if this codes for the same amino acid as this, the protein will remain the same. But if this codes for a different amino acid as from this, then it means that the protein will change. So it means that if you come here, GAA, it is leucine, while GAA, GGA is proline. It means that change of this to this, change of this to this will result in a different amino acid being coded from leucine to proline, which will result in a different protein. Change in CUU to CCU will result in a different amino acid coded. That is from leucine to proline. From leucine to proline, which will result in the structure of a protein. Yeah, I think I'm going to stop here. Yes, I'm going to stop here I'm going to stop here. When I come back, I will finish this paper from 2.3. Thank you very much. Please don't forget to subscribe. If you have many questions, drop them, or you can send them on WhatsApp. We will answer them before or with immediate effect. Thank you very much. Let's continue with the question 2.4. It's about genetics. We know that many students find challenges with the genetics. Question 2.4 from National Senior Certificate 2022, NCS 2022. The diagram below shows the inheritance of flower color in snapdragon plants. The two alleles controlling the flower color are red and white. It means that because the two alleles are capital letter, this is a capital letter, and also this is a capital letter, it means that both of them are dominant. So if both of them are dominant, here it's showing us that you have a dominant red crossing with white, and it gives you pink. When you have red and you have white, we produce pink. So pink is neither red nor white, but is an intermediate between the two. So the type of dominance which is there, the moment you get an intermediate, neither of the parent color produced in the offspring, it means that that kind of dominance is called incomplete. It means that red is neither dominant over white, and white is neither dominant over red. An example is different presidents of different countries. When they sit on one table, all of them, they will talk about issues affecting them equally. So it means that neither of the president of country A nor the president of country B is dominant over the other. Why? Both of them or all of them are presidents. So neither red nor white is dominant over the other. So when they combine, they produce an intermediate. So let's go to the questions and we see. They are saying that state the type of dominance shown by the snapdragon plants. So we have seen that it is incomplete dominance. If it was complete, then we would have got either red or white in the offsprings, or both. Red, some of the organisms are red and some of them are white. That is complete. If it was codominancy, we would have got a mixture of red and white. But now we get an intermediate, therefore it is incomplete dominance. Give a reason for your answer. The answer, we have explained it, that you produce an intermediate, or an intermediate color was produced. A gardener crossed 2 pink flowers. Use the genetic cross to show the ratio that is expected of when you cross 2 pink flowers. 2 pink flowers. So it means that if both of them are dominant, definitely this is going to be capital R, capital R, capital W, capital W. Yes, they produce one gamete from this side and one gamete from that side. So this one is going to produce a capital R. This one is going to produce a capital W. So it means that this crosses with this, you produce this. And RW is an intermediate of red and white, which is pink. So it means that when you're talking about the genotype of pink, we have to use this. So what are you supposed to write? Okay. We are saying that now, the first thing you need to do is you say, because it is a second generation we are crossing, therefore it is advisable to use P2. So it's going to be P2. You tell us the P means parent. So P2, parent 2, because we have already parent 1. So now pink and pink, it means that that's P2. Yes, if it's a second generation, we use P2. P2, yes. We can say F2, second Fourier generation. We mark this as ones, like, so give you one tick there. So don't forget, if you write P2 or P1, F1, it must be, yes, it must be P2, F2, P1, F1. If you write one, you don't write the other one, then you get it wrong. For example, P2 and don't write F2, you don't get a tick. So now P2, you tell us the phenotype. The phenotype is pink. We cross it with another pink flower. The X must be like this. It must not be like this. No, the X must be like this. Then you go to genotype. What's the genotype? We have seen the genotype here. That is capital W, capital R, or capital R, capital W. Therefore, we're going to have to say here, it is capital R, capital W, cross with capital R, capital W. Now, these must separate under meiosis. So you write the word meiosis. So meiosis, you form the gametes, capital R, capital W. Here, capital R, capital W. So we call these ones gametes. Gametes. Don't leave these gametes naked. In most cases, I always tell my students not to leave these gametes naked. They're not at the beach. All right. We circle them. We circle them. We circle them. Even if you call them, still a tick, but I prefer to circle them. Why? So that we can differentiate them from the genotype. And always know that there must be a space between these two gametes. Don't combine them, because when you combine them, then it will look like it is a genotype. There must be spaces there. All right. Now these gametes, they're supposed to fertilize each other. Don't use the word fission. We won't give you a tick. Fertilization. Fertilization. When fertilization occurs, you can use a Punnett square, or you can use crosses. Since it is a monohybrid cross, you can use crosses. For example, this goes with that. So I'm going to have capital R, capital R, and now this with that. So it's going to be capital R, capital W. Now this with that is going to be capital R, capital W. Then this with that. So it's going to be capital W, capital W. Yes. So this is what we call fertilization. So these are the offsprings of the parents. So F1 offsprings. What are they? They said snapdragon. It means that those are the offsprings or the snapdragons. But what was the question? The question is saying, use the genetic cross to show the ratio of the expected phenotype in the offspring. So we are looking for the ratio of offspring. Okay, let's look at the ratio of the offspring. Now, capital R, it is red. So I have red. How many are they? It's 1, comma. I have capital R, capital W. It is pink. Capital R, capital W is pink. So I have 2 pink. And then now I have capital W. It's white. I have 1 white. So basically I have 1, 2, 2, 2, 1. So that is it. So now, when you are looking for a mark, when we mark, we are looking for ticks, and then sometimes we put what called a compulsory mark. So in case we come here, we do this, we do, yes, this and that, we give you a tick. Phenotype, we give you a tick. It must have a cross. Genotype, we give you a tick. It must have a cross here. If it doesn't have a cross, the tick might not be given to you. So we come to the gametes, we give you a tick, and then we have for crossing, and you get the offsprings, we give you a tick. So because the question is saying give the ratio, so this one might be a compulsory mark. It might be a compulsory mark. So we give a tick, but we put the word C there, which means that it's compulsory. There is one mark short, which is meiosis and fertilization. Give you a tick. So how many marks are there? It's 1, 2, 3, 4, 5, 6, 7. And then in most cases, we give you 6 marks. And also sometimes cross, we can give you a tick there. So maybe we can say that this question is 6 marks. But how do we count? When you are counting, first count this. However much you get all of them, like you have all of this. Without this now, for this case, you won't get 6. You have to be with all of them. You have to be with all of them plus this. Or you have to be with this plus another 5 so that we make them to be 6. So that's how we mark the genetic cross. This question, it is 2022 Department of Basic education in CS. Another question is about evolution. Evolution. Here they're saying that diagram A and B show the ventral, that is the bottom side of the skull, of the skulls of two organisms. The diagrams are not drawn to scale. It means that they are not, that's not their size. So now asking, which diagram represents the skull of bipedal organism? Let me first explain the word skull. Skull. Some people, they confuse the word skull and cranium. Cranium is where the brain goes. The skull involves even the jaws. So when you are meaning where the brain goes, please don't use the word skull, use the word cranium. So skull is the whole thing, the head, including the jaws, the teeth, and everything. But when you talk about cranium, please, we are talking about where the brain goes. All right. Now, which diagram represents the skull of bipedal? Bipedal, it means that organism is walking on two limbs. If the organism is walking on two limbs, it means that what shows we have three factors which you can identify whether the organism is walking on two limbs or not. The first one is the position of foramen and magnum. The second one is the shape of the spine, if it is S shape or C shape. S shape, it means that it allows flexibility for the organism, so therefore it is bipedal. And then C shape, the flexibility is not there, therefore it works on four limbs, quadrupedal. So the pelvic girdle for the case of bipedal must be short and wider to support the upper weight and allow easy movement. The other one, it must be small and narrow. Then foramen magnum for the bipedal organism, it must be in the forward position. That's how we want it, in the forward position. While the foramen magnum, it must be in the backward position for the case of the organism which works on four limbs. Why is it supposed to be in forward position? Forward position so that the spine can connect to the skull vertically. And then now the skull is going to face straight in front. Straight in front. Hence, the organism is going to walk on two limbs. So if you look at this, it's a little bit in the forward position. Don't say in the center. Center of what? No. Even this side can be center if I count from I measure from this side to this side. So it's going to become the center. So the center, we say in forward position. Well, this one is a little bit backward. So it is not in the forward position like this. So it is backward. So it means that this is a bipedal organism. This one is the quadrupedal organism. So they're saying, give one visible reason for your answer. So the answer here is B. If the answer is B there, therefore it means that give one reason. It's because the foramen magnum is in the forward position. So it is in the forward position. And then they're saying that tabulate two visible differences between the upper jaw. Now here, they're looking only and specifically the upper jaw. These jaws which you see. So don't talk about other parts. The upper jaw in diagrams A and B. When they say the word tabulate, that's a mark. The word tablet, even you can see that is 5 marks. So when you draw a table, please make sure that you draw a correct table so that you get all the marks. So you write here, for example, skull A and then skull B, for example. Make sure that you close the table. You lose marks here. Ne? Don't just draw a T and then you write. No. If you draw a table, make sure that you put the heading. Ne? Please close. Don't just draw it like that. Please close the table. Close the table. It doesn't cost you anything for you to close the table so that you don't lose a mark. Be neat when you are writing. All right, now let's compare. Skull A, skull A. This is skull A, skull B. So number one, They're saying the upper jaw. Let's look at these teeth. This and this. The canine here, they are big, meaning that they are well-developed canines. Here, the canines are less developed, meaning that they are small. Number two, this one has a rectangular palate. Here, it has a U palate. Ne? It's like a C or a U palate. Here, it is rectangular palate. So, those are some of the differences you can talk about because they are talking about the jaws. So the palate here is rectangular palate. Here it is U shape, or the jaw, they are U-shaped, and then here, yes. And then they are saying that so you get 2 marks for the difference and 1 mark for the table. Then they're saying explain the significance of the shape of the spine. I talked about it. That is associated shape of the spine, significance of the shape of the spine that is associated with the skull B. What is skull B? Skull B, we say that it is walking on 2 limbs, and the shape, if you look at it, it is S shape. Because they didn't talk about which shape is it, then mentioning the shape could make you to earn a mark. Therefore, the shape is S shape, yes, S shape. But what is this S shape for? It allows flexibility of the organism, so it makes the movement a little bit easier. Then, in most cases, things related to the graph, I only point out what is very important and the most likely questions which are likely to come when they bring a graph, because so that there is no cramming about the graph. So now, if they ask you to describe, when they say you're supposed to read the whole question. Herbicide is a chemical which kills a plant. Glyphosate is used to control the weeds in maize fields. The herbicide kills the weed, but it cannot kill the maize plant. The herbicide kills the weed, but it can also kill the maize plants. It has been found out that weed developed resistant to glyphosate treatment. Scientists carried out an investigation to determine the development of the glyphosate resistance in the weeds. The weeds were treated with the same concentration of the glyphosate each year from 2009 to 2016, and the percentage of the glyphosate resistant weed in the field was recorded every year. Okay. Results as follows. You see that glyphosate resistance, as the year goes on, the resistance increases, but 2015 and 2016 the resistance became constant. So now I'm saying this could be a change in the glyphosate resistance over a period of year. It means that here, whenever they ask you to describe, you tell us what is happening. Because if you draw this on the graph, not a bar graph, just a normal graph, you will see that it will be continuing, and then it forms something like this. So there is an increase from 2009, 2015. There is a steady increase, né? Yes, in the resistance of the weed over the herbicide, and then it remained constant between 2015 to 2016. So it remained constant. So when you say description, you tell us what you see. Don't give us the reason why there is that increase. That is description. When they ask you to explain, then you tell us why, you tell us that there is an increase, and then you give us a reason why that increase is there. So that is what we call explanation. But if they ask you to describe in science or in here, you just tell us the shape of the what? Of the graph, or the trend, how it moves. That's what they want. That's why you see that the marks, even they're not that a lot. Then they're saying that calculate the percentage increase in the glyphosate resistant weeds from 2010 to 2015. Show you're working. Here, whenever they ask you to calculate the percentage increase, whenever they ask you to calculate for the percentage increase, this is what you are supposed to do. Percentage increase is equal to the final percentage increase is equal to the final figure, the final value minus the initial value over the initial value times 100. Yeah, that's it. So in this case, they say that from 2010 it is 20, and the 2010 is the initial value, and then to 2015 is 65. So I'm gonna say the percentage increase is gonna be equal to 65 minus 20, yes, divided by because the initial, it is 20, then times 100%. So the answer you get is gonna be the percentage increase. So that's how we calculate the percentage increase. So this is a rough thing how you can answer questions when you really sit in this paper or any paper you're writing. You can really have no questions which are challenging. You are not going to see any new questions. All the questions you're gonna find in the paper are always the questions we ask you every day. Those are the questions you're gonna find in the paper. So there is no new questions because we don't have any new science which has come in. So the curriculum remains unchanged for a period of years. So no new questions. Don't expect new questions in your exam. So please try and focus on, we have the same format of setting, so we cannot set outside the examination guideline. So scientists isolated the gene from for glyphosate resistance from the weed and used it to genetically modify the maize plant. Okay, explain the economic benefit of making the maize plant resistant to glyphosate. Because the glyphosate, now look, they are saying that herbicide is a herbicide which kills plant. Maize is also a plant, and then we use it to control weed in the maize plantation or the fields. So we have to make sure that this glyphosate cannot kill the maize. So the reason why they make this maize to be resistant to the glyphosate, so that when you apply the glyphosate cannot kill the maize what? The maize plant. So that is the idea behind it. So I'm not saying that this is the exact question which is going to come, but when they give you such question and you don't have any idea, you can go back in the passage, most especially these questions which are involving of reading, go back, read again, and then identify. Then they're saying that draw a bar graph. Before I go to the bar graph, can you identify the independent variable here? You can identify the independent variable here. So is the weed dependent on the herbicide, or the herbicide depend on the weed? It means that the weed will depend on the herbicide over a period of years. That's why here you see that over a period of time or years. That's why you see here that glyphosate resistance, the resistance will accumulate based on the period of years, meaning that this herbicide will be resisted based on the period the weed has stayed in contact with the herbicide. Therefore, the resistance depends on the contact time it has stayed with the herbicide. Hence, resistance becomes the dependent, and then the period of the contact becomes the independent. Okay, the bar graph. Now here, when you talk about the bar graph, you need to know the things we are looking for, so that when you go to the exam, you don't miss any question concerning about the bar graph, concerning about the graph. The first thing you need to know is the caption. Sometimes some teachers don't explain, they just mark, they put a T, they put a C, they put a S. They don't explain this to their students. But what does it mean? How do you answer this question so that you don't skip any mark? All right. So now they're saying that the bar graph should illustrate the results of investigation in the first 4 years. So, okay. Now, I'm not going to draw the actual graph, but I'm just going to talk about how you can draw this bar graph without stress. So years are always an independent variable, meaning that this is time. Time is always an independent variable. So independent is always on x-axis. Independent is always on the x-axis. And then the dependent one is always on the y-axis. I always tell my students that if you forget which one is on the x-axis, which one is on the y-axis, you just write here the word ID, your ID. Ne? Yes, ID, which means that you know the ID you are using. Independent, dependent. So this one is going to go on the x-axis. Then you here put ID, X, Y. So this one is going to go on x-axis. This one is going to go on the y-axis. So that's how you can remember the x-axis and the y-axis. So you just do this small box. I repeat. Ne? You say ID, X, Y. So meaning that ID, independent variable on x-axis, dependent variable on y-axis. The first column on x-axis, the 2 or 3 or 1 goes to the y-axis. All right. Now, if you are drawing a bar graph, make sure that you draw a straight line. Don't mind about mine. Straight line. This is how we mark. You write a caption. Never forget about the caption. So we give you a tick for the caption. Then after that, you write the X, you write the Y here, you write the X-axis here. But when you're putting the value here, for example, in this case, they are saying yeah, and then the percentage, the glyphosate resistant weed percentage. So it means that the resistance is going to be here. Yes, the resistance is going to be here. And then units, it is in percentage. Then the time is going to be here, which is year. And then the years, they don't put anything. So if you just say time in years or just year, then it's fine. It means that now they're going to give you another tick for labeling the what? Labeling the scale. Ne? Not labeling the scale, labeling the axis. So if you label the axis X, they can give you a tick, or Y, they also can give you a tick, or they can combine the two. Now, you go to, after that, you go to the intervals. Please don't start with a 0 here, yet you want to start. If you come back here on the, you'll see that is 10, 20, 32. Okay, this one, it's fine. You can start with a 0. But if it is starting from maybe 100, and then you go to 120, you'll find out that some people, they start here 0, 100, 120, 140, 160. So the difference between 0 and 100 is not the same as from 100 to 110. So if you start from 0, make sure that, and you say a difference of 10, it must be 10, 20, 30, 40, 50, 60. It must be consistent. If you don't do that, you're going to lose a mark. And then now, you're going to have to draw your bar graphs. It must be straight. Don't forget that these bars must be straight. It must be straight. It must be straight. Ne? So now, the distance between these bars, I'm saying this is a rough distance between this space and this space must be equal. It might not be exactly the same because you don't use a ruler when you're marking. No, but when you look at with your eyes, you see that these distances are the same. So it must be equal. These bars, the size must also be equal, so that we give you, if the scale, the interval is the same, we give you a mark for the scale. Yes. If you have drawn the bar graph, not a histogram whereby the bars are connected to each other, then we give you correct type of the graph. We give you a correct type of the graph. We give you the mark for the caption. Then we give you a mark for labeling the axes. We talked about it. Then we give you a mark, maybe 3 of them. We give you a mark for plotting if you have plotted this correctly. So that's how we mark the bar graph or any other graph of any kind of nature. So basically, that's how we mark. Then the last question is natural selection and also out of Africa hypothesis. Still here, I'm not going to answer the actual, like the way it is here. I'm just giving you the ideas how you can answer these questions without stress. Because when you are bringing the question, we can bring it in a different version. Yes, we can bring it in a different version so that you don't lose those marks. So when you modify, where do you modify and how do you modify it? A population of lizards on the island A were well situated to feed mainly on insects. So it means that these were feeding on insects. Scientists moved the 5 adult pair of the lizards species to the neighboring islands. They are saying the island A, island B, here they reproduced and a new population formed. They're saying the island B has large supply of plant with the fibrous leaves and fewer insects. It means that now the food has become scarce. So this one is going to become a problem. Yes, exposure to this insect new environment may have caused the lizard to undergo evolution. Okay, they're saying 63 years later, scientists returned to the island B to conduct a further investigation on the lizard population there. They observed that the jaw size of the lizards had increased, and scientists also analyzed that the stomach content of the lizard found that it was mainly plant-based. They confirmed that the 2 populations still belong to the same species. They confirmed that the 2 populations still belong to the same species. Describe how scientists could confirm that describe how scientists could confirm that there was a change in the jaw size between the lizards of island A and the lizards of island B. So the question might not come like this, explaining this, but it can come in a different version. So we shall go and then see exactly where we need to have the answers from. So basically, what the scientists could use to confirm or could confirm that there was a change in the jaw is, this is an obvious answer. It means that they had to measure the lizards on the island A and the lizards on island B, and they check whether the jaw size is the same, and they can compare the differences if the difference is there. Then explain how the large jaw of the lizards on island B would be structurally suited to eat the tough fibrous. Explain how the large jaw of the lizard on island B would be structurally suited to eat the tough fibrous leaves. Yeah, this could be about the jaws being strong, meaning that the muscles are strong enough, and also the teeth, so that they can have a strong bite, and then they can chew that food. It means that the muscles, to do that, it must be strong, so that the force of grip is high, and then they are able to chew this kind of food. How did the scientists determine that the 2 populations of the lizards, both on island, still belong to the same species? Don't forget that a population a species, these are organisms that can interbreed to produce a fertile offspring. So if they have the two populations and they try to allow them interbreed and they produce a fertile offspring, therefore they can confirm that these are still the same species. But if they cannot interbreed to produce a fertile offspring, then automatically they are not the same species. But if they could interbreed and produce a fertile offspring, therefore it means that they are still of the same species. Explain the possible effect that evolution of the lizard has on the biodiversity. Explain the possible effect that evolution of the lizards has on the biodiversity. Here, it could be, it can increase the biodiversity or it can reduce the biodiversity. So basically, those are the two ways. If it forms a new species, then automatically it has increased the biodiversity. If it takes away an existing species, then it forms another species, then it means that it is decreasing the biodiversity. However much is creating, but it's decreasing because the other one is no longer existing. Or it could be 3 species, and then because now the food has come in a different angle, the 3 species, they modify into 1 species, or they modify and the other ones, they become extinct. They become extinct, meaning that now they have also decreased the biodiversity. Use the Darwin theory of natural selection to explain the evolution of lizards with large jaws. Use the Darwin theory of natural selection to explain the evolution of lizards with large jaws. Here, the principle is the same. The principle of natural selection, whatever you explain, use the same principle. The first point you have to talk about is variation. There was variation among, now you have to tell us what they gave you, among the lizards, those with the large jaws and those with the smaller jaws. Because the jaws are used to bite or to eat this tough food, therefore, those lizards with big jaws, they could eat enough food, tough food, therefore they survived and reproduced in large number. Those with the small jaws, they could not eat enough food, therefore they died in large number. This is called natural selection. Yeah, previously we are giving a tick on natural selection. This is called natural selection, but the chances are high that we're no longer giving this marking point of this is called natural selection. But put it there, you never know that maybe we can review about it. This is called natural selection. So those with big jaws, they pass their genes to the next generation because they reproduce in large number. After many generations, all the lizards have big jaws. There is variation, I'm repeating, there is variation among the lizards. Those with, you tell us the variation. Those lizards with large jaws and those with the small jaws. Those with the large jaws, they could eat the tough food. Yes. And then they survived, they reproduced in large numbers, they passed their genes to the next generation. While those ones with the small jaws, they could not survive, they could not eat enough food, therefore they died in large numbers, and this is called natural selection. Those which survived, they passed their genes to the next generation, and after many generations, all the population of the lizards were made up of large jaws. That's how you can explain using Darwin's theory of natural selection to show how this evolution of large jaws came about. Lastly, on this paper, please, you should never go to the paper without knowing these questions. The out of Africa hypothesis, which states that modern humans originated from Africa and migrated to the rest of the world. Don't forget the word modern. However much they might not give it as a ticket marking point, but in most cases, we criticize when the word modern is not there, because not all humans are modern. Homo habilis were humans. That's why you call it Homo, which means a man. Homo erectus were humans. That's why it's called Homo, is a man. Homo sapiens is a human. That's why you call it homo, which means a man. So Homo habilis, the upright, the handyman, Homo erectus, the upright walking man, Homo sapien, the wise man. Those are, all of them are humans. That's why we are referring to modern humans. So please put the word modern humans, Homo sapiens, which means the wise man. I don't mean the school, I mean the man, you who is listening. Someone's going to ask me, what about us who are ladies? Yeah, is a man in science. All right. Hey, they're saying that name the family to which modern humans belong. They belong to a family called Hominidae. Hominidae. So they belong to a family called Hominidae. Yes. That's the family to which it belongs. They're saying that which genetic evidence is used to support the out of Africa hypothesis. We have two genetic evidences, but we only teach one. We have what called mitochondrial DNA and Y chromosome. But now we don't talk about the mitochondria. We don't talk about the Y chromosome. Only now the syllabus or the curriculum is talking about the mitochondrial DNA. So the evidence we give you here is mitochondrial DNA. Describe how fossil evidence is used to support out of Africa hypothesis. I've been telling these kids that these are the same questions we ask you. And if you check the book, our book we use, you can download even yourself. That's exactly how we mark exactly. This is the same way how they ask you this question. This book was released a long time ago before even these papers are being set. All right, let me answer. You have to talk about the oldest fossils which were discovered in Africa. For example, you have to talk about that Adipithecus remedus was discovered in Africa. You have to talk about that Australopithecus. Among Australopithecus species, we have Lucy, that is Afarensis. We have Taum child, that's Africanus. Mrs. Place, Africanus. Little fruit, Africanus. Carabo, Sediba. Ne? All those Australopithecus species were discovered in Africa. Homo habilis, also in itself, was discovered in Africa. That is a handyman. Now, when you reach here, you add the word the oldest. The oldest species, the oldest fossil of Homo erectus and Homo sapien were also discovered in Africa. That's what you want. So you may not talk about all the Australopithecus species, all of them. You can just talk about one or two. So Adipithecus discovered in Africa. Australopithecus species discovered in Africa. For example, Taum child, Mrs. Place. Then you'd go to Homo habilis, discovered in Africa. Homo erectus, the oldest, Homo erectus and Homo sapien, discovered in Africa. Let me show you what I'm trying to say. This is what I'm trying to say. Evidence to support out of Africa hypothesis. There are three: the mitochondrial DNA, the cultural evidence, and the fossil evidence. This is what I'm trying to say. That fossils of Adipithecus were only found in Africa. Fossils of Australopithecus, e.g. Carabo, Little Foot, Taum child, Mrs. Place, all were found in Africa. Homo habilis were only found in Africa. Oldest fossil of Homo erectus and Homo sapiens were discovered in Africa. This shows that modern humans originated from Africa and migrated to the rest of the world. You see that even I highlighted the key points which you need to know when you're writing in your exam. Yes. So as I'm winding up, I will recommend you to use this book. Go to our website, download it, free of charge. If you want to be printed, that's a different story. But go use these resources. I took time to write this book and I wrote it with care so that every child of this country in South Africa must pass. You don't have a right to fail. You have a right to pass. You must demand for your rights. What is your right? The right of passing. Please, never ever demand for the right of failure, because you have the ability for you to pass, not the ability for you to fail. I wish you the best. Make sure that you go through these papers. You will make it. Thank you very much. Don't forget to subscribe, like, and share to others so that they also benefit in this before they write their final exam. Thank you very much. M.Saidi, as usual. A student asked me a question: how do I use my book of distinction material to get a distinction? That I said that whatever question is being asked is being indicated in that book, and asked me that I should use 2022 National Senior Certificate Examination to answer that question. So let me show you exactly as we are comparing the question versus where do I find the answer in the booklet. For this case, I will only focus on questions not related to objective. So on the side of the objective, I'm going to be a bit faster. Let's start with the section A. The component of DNA molecule that provide the code for protein synthesis. Which component of DNA molecule that provide the code for protein synthesis? So what causes this are the nitrogenous bases. What are the nitrogenous bases? We are talking about the thymine, the guanine, the cytosine, and the adenine. These are the nitrogenous bases which are supposed to be organized together to form a gene. So these are the ones which bring about a protein. So the answer is D. Then 1.1.2, during which stage of meiosis do spindle fiber begin to form? So the spindle fiber always begin to form under the preparation phase, which we call the prophase. So it means that the answer is going to be A. I'll explain more when we go to other sections. Then they're saying that an individual that has Down syndrome in the karyotype, there is an abnormal number of chromosome at chromosome pair. So Down syndrome, whenever you talk about Down syndrome, yes, so your mind must go to chromosome number 21. Whenever you talk about Down syndrome, so you must think about an extra chromosome on chromosome number or chromosome pair number 21. So the answer is going to be C. So don't forget that these objectives, each objective is 2 marks. Then they're saying that an individual that receives an identical allele from each parent is described as, how do you describe an individual that receives an allele which is identical from both parents? For example, you have the parent is capital T, small t, and then another parent is capital T, small t. So now, if I receive this and this, how do I call this? So now, if the baby gets small t, or this and this, and the kid is capital T, for example, yes, it means that these 2 alleles are the same. These 2 alleles are the same. So how do we call that individual which receives those 2 alleles? So because they are the same is homo. So it is going to become homozygous, because you are describing a zygote, yes. So that zygote has the same kind of allele. So the answer is going to be C, meaning that homozygous. Then the next question, they are saying that the plant species that has a diploid chromosome number of 12. So if a plant species has a diploid chromosome number of 12, yes. Which of the following is a haploid chromosome number for this species? It means that if it diploid, di means 2. Di means if it is diploid, it means 2. That's why it's called 2 sets of chromosomes.